imaging is one of the most visual ways of measuring biology, right? So you actually get to see what's happening, and that's one of the strengths of this technique as well, that you get this dynamic description. So when we look at those videos, we would see fluorescently labeled neurons in living C. elegans embryos as they are developing. And so you can look at individual neurons, the decisions they are making as they're born, as they grow out, as they're placed in the brain that's also developing at the same time. So you can look at these single neurons and their decisions in the context of the developing brain, which we think is really cool. When a neuron grows out, its ending is very dynamic, so you would see certain structures forming, and there are points where you can see the neuron making its decision. So when a neuron is initially growing out, you can see that there are like these small extensions that are forming, and then one of them goes on to become the final extension or the axon, and so you can actually see those decision points. What we are able to do looking at those videos is that we would find snapshots where we are seeing some critical points where the two partners are meeting each other, and so you will see that there's a point where the two partners have just met, and then we see these like crazy event where like the two neurites sort of move as a whole and zip up, which is not traditionally how neurons are thought to be positioned. So traditionally it's thought that neurons mostly grow out from their tip, and that's what determines mostly their trajectory. But here we see this event which is sort of new and not something that we expected, so it was counterintuitive. It was really cool, yeah.